Hello, welcome to Intro to Psychology, Chapter 3. In this video, I will be discussing the transmission of a neural impulse within a neuron, the chemical changes that occur in order for a neural signal to propagate down a neuron and allow our brains to communicate with the rest of our body. So an important term to know in this step is the action potential. The action potential is a change in electrical potential or voltage that occurs when a neural impulse travels down an axon. And it is described as an all or nothing response. And we'll see what that means in a moment. Just as a quick review, remember that sodium ions are highly concentrated outside of the neuron, and potassium ions are highly concentrated inside of a neuron. And it's the movement of these ions that allows the action potential or the neural signal to go through down an axon. So there are five basic steps in the action potential that allow or explain the movement of a neural signal in the nervous system. These five steps are very, very important. Make sure to know these for the quiz. So the first step of the action potential, you'll see that when the neuron is at rest, it's at a resting potential of -70 millivolts. So it's right here when there's nothing going on, when there's no neural signal being propagated. Then, eventually, once a signal does come from another neuron, what happens in step number two is that sodium channels begin to open. When the sodium channels open, the positive ions rush in to the neuron. So remember that sodium was highly concentrated on the outside of the neuron. And so, because of the concentration gradient, as well as the opening of channels due to the voltage, the sodium ions rush in. So the influx of positive ions means that the inside of the cell becomes less negative or it becomes more positive. So it goes from a level of uh, 70 millivolts up to about -55 millivolts. And if it reaches that threshold, then an action potential is started. So once the influx of positive ions brings the action potential to this threshold, then all of a sudden many more sodium channels open, and sodium ions just rush in, and the inside of the cell becomes more and more and more and more positive until it reaches this peak of about plus 30 millivolts. And at this point, potassium channels open. So remember that potassium is highly concentrated on the inside of the neuron, and so when potassium channels open, the positive potassium ions rush out of the cell down their concentration gradient, and make it so that the inside of the cell becomes more negative. So here, sodium channels are open, and it's becoming the inside of the cell is becoming more and more positive, more and more positive. And then here, potassium channels open, so positive ions rush out, and it becomes more and more negative, more and more negative. And then in the fourth step of the action potential, the eventually the sodium channels close and the potassium channels remain open. And so the potassium ions are still rushing out of the cell, making the inside of the cell more negative than it was even at rest, even as it was in comparison to the resting potential. And so we call that hyperpolarization. So in depolarization, in this kind of rising action, you have a positive pole and you have a negative pole. The outside of the cell is very positive and the inside of the cell is very negative. During depolarization, they become more similar. The inside of the cell becomes more positive and it becomes more like the outside of the cell. So it's being depolarized. The poles that were at their extremes are being brought closer together. So they're being depolarized. And then repolarization, they're being repolarized. The outside becomes more positive and the inside becomes more negative. And then during hyperpolarization, it becomes even higher, a greater extent of polarization than was present at resting potential. And then finally, in step number five, eventually those potassium channels close and the neuron returns to resting potential. If you visit neuroanatomy.ca and look at the Intro to Neurophysiology interactive module, you'll see some great images and some great interactive modules under the axon portion explaining this in detail and showing you diagrams of the channels opening. I mentioned in a previous video that there are two main types of cells in the nervous system. The second main type of cell in the nervous system is glial cells. And glial cells play really just as important of a role as neurons, even though neurons typically get most of our focus. And glial cells are cells that help with the transmission of the nerve impulse by providing structure, insulation, and protection to neurons. They protect against pathogens, they provide structure and support in particular areas of the nervous system, and there's almost as many glia as there are neurons in the human brain. One important example of glial cells functions is myelin. So myelin is a substance that coats the outside of axons that is created by glial cells in the central and peripheral nervous system that allows for insulation and allows the neural impulse to travel faster down the axon. So if you take a look here in this diagram, this is an unmyelinated neuron. It doesn't have any myelin on it. And you can see that the neural impulse is moving okay, at kind of a slow pace down the axon. Whereas here, in contrast, uh, these cells right here covering the axon are myelin cells that provide insulation. And what they do is they block the action potential from occurring, except for at specific points. These specific points are called the nodes of Ranvier, named after the person who initially identified them. And so the action potential can only, the ion channels can only open here and here and here and here and here. And so the neural impulse travels much faster down axons that are myelinated in comparison to axons that are unmyelinated. And we call this kind of propagation of the neural impulse saltatory conduction, in the sense that it jumps from one node to another rather than going through every single ion channel possible that an axon has. And myelin play a very important role, and it's seen in several demyelinating diseases such as multiple sclerosis, where in MS, the myelin cells are attacked by the immune system. T cells from the immune system somehow mistakenly identify myelin as foreign pathogens and begin attacking them and degrading them. And what that does is, as you saw in the previous slide, it slows the neural signal from being propagated throughout the nervous system. And that results in a variety of symptoms throughout the nervous system, as well as affecting other bodily systems, such as cognitive impairment, difficulties with speech, various pain symptoms throughout the body, difficulties with bowel and bladder incontinence. People with MS most likely end up walking with a cane, or losing the ability to walk early on in the disease process. And it's such a variable disease, because any degradation of myelin anywhere in the nervous system can result in all kinds of different problems for a person's functioning. So in the next video, I'll be discussing the transmission of the neural impulse between two neurons. How do neurons communicate with each other to give rise to all of our mental functions?